Never a dull moment at the bucket courses. Welcome to the bucket courses, the perfect place to learn new things, meet new people, see interesting things happen. My name is Barb Lease. I'm glad to see all of you, and here are some things you need to know before we start. Masks are welcome, but not required. Please silence your cell phones. Turn on your T-coil if you have one. If there's an opportunity for questions, we'll come around with the mics. Please speak directly into the mic. Coffee and cookies cost about $2 a person. If you're able, consider putting a little money in the big green bucket. If you're able, please put your chair up after the program on the dollies provided at the back and the front. And now, finally, for the main event. Professor Gene Wubbels earned his BS at Hamline University and his PhD at Northwestern University, where he was an NIH pre-doctoral fellow. He was professor of chemistry at Grinnell College from 1968 to 1992, served as a program director at the National Science Foundation, was a dean and professor at Washington College, and served as an administrator and chemistry professor at the University of Nebraska at Kearney from 1995 to 2013. He and his wife Joyce are enjoying their retirement in Grinnell. This morning, Professor Wubbels will shine a light into DNA research and how it has affected medicine, law, agriculture, drugs, vaccines, and the growing biotech industry. Please join me in welcoming Professor Gene Wubbels.
Thank you all. I guess the right way to put this first question is, anyone who can't hear me, please raise your hand. Seeing none, thank you. Nice to all of you to come out this morning, the second day of spring. Today, I would like to tell you a story about a history of synthetic DNA. And I say by a scientific bystander, this will be pretty much a personal history. There are printed histories of this out there in the literature, but mine will be personal for reasons I'll explain. Early in the COVID pandemic, I realized that there was some pretty fancy chemistry going on behind these things, and I didn't know what it was. Here are some examples of what made me curious. I knew that there were billions of doses of these vaccines that had been made. They're impressive achievements developed in a very short time. And the diagnostic tests that I had after I caught the virus were also puzzling to me, because they seemed to me to be just amazing. They're very highly specific, and as well as very fast. So then I got to wondering about all these drugs I'm seeing advertised on TV. And here's a list of 6 of them that I noticed in the last few weeks. And their manufacturers. I looked them up and found out that all of these are a thing called a fusion protein, with the exception of this, which is another kind of protein. And I'll tell you what that is later. That's for these diseases. And they're another puzzle. They're all made by something called recombinant DNA in CHO. What's CHO? CHO is Chinese hamster ovary cells. My wife Joyce was curious about that. Maybe you are too. I'll explain as best I can later. So then I thought, I wonder if there's any other things of this kind. So I looked up this recombinant DNA in Chinese hamster ovary cells, and I found that it's been around since 1987. That was the first drug that was used, that method of synthesis. This Epogen is one that I'll tell you about in some detail later by the Amgen company, another important part of the talk. But this is quite a list up until 2006. And I assure you that there are many, many more of these that have come since then. Here are some social contexts that you might have noticed the words DNA in it. don't know if you know that there's DNA sequencing involved and another thing called synthesis, which this talk is about. But here are the 5 areas I thought of and what's going on with synthetic DNA and all these different things. So the discoveries that all these things depended on synthetic DNA hit really close to home for me, at least scientifically speaking. It's because I did my PhD research at Northwestern University in the laboratories of Robert Letsinger, who's shown here. This was a picture taken about 30 years ago with his wife Dorothy and their dog Tuppence. And this was a wonderful family to get to know. They were hospitable to the group. We had frequent group meetings in their house, and they served us dinner occasionally. So very accommodating people. Most of the people in the Letsinger lab, however, were not doing what I was doing. I was doing synthetic organic, well, excuse me, not synthetic. I was doing organic photochemistry, and most of the group was concerned with something very different. They were trying to make synthetic DNA. This was in 1965. So how did I learn about synthetic DNA? Well, because I had to sit through group meetings every 2 weeks in which all the DNA guys would present what they're doing in research in DNA, and then they, of course, had to listen to our photochemistry talks too. But I got a real fetching up in synthetic DNA. I knew what our group was trying to do. I knew what a number of groups around the world were trying to do to make synthetic DNA. When I looked into the research on this, I realized that there was another important actor in this. This fellow here, who's pictured about 30 years later than that earlier slide. This is about 2 years before Bob died. That's Marvin Carothers. He was in the research group when I was there, and he was one of the DNA guys. Carruthers went on to have a distinguished career, and he's still at it, at the University of Colorado. And when you try to look up the history of synthetic DNA in the scientific literature, you'll find out that you only need to know 2 names, Letsinger and Carruthers. That's quite astounding, because all around the world, a lot of people were trying to make synthetic DNA, but the ones who succeeded and created the methods of doing it that are now used worldwide are these 2. So with that tutorial that I had, I thought I ought to do a history of this. So after all, I had a lifetime of trying to explain chemistry to people that didn't know very much chemistry, and that here was this fantastic story that plopped right in my lap, and I thought, yeah, I ought to give a talk on this. So I looked up one of the histories in the literature by these guys, and they said at the start of their article that if the 20th century was the century of the atom, that's technologically speaking, what we are witnessing with DNA could very likely become known as the century of DNA. Well, wow. So let's take a look at the structure of DNA. And I ask you to bear with me on this. I won't go very deeply into this, but we do need to have some idea of what these things are and how they interact in order to talk about what they're doing. There are 3 classes of giant organic biological polymers called DNA, RNA, and protein that do most of what our cells do for chemistry business. DNA, long, very long, thousands of units long, single or double strands of this stuff, deoxyribonucleotides. Those are the monomers. We're going to talk here about monomers and polymers. I already mentioned that. A polymer is simply a giant molecule that's made up of many parts. Often the parts are the same or similar, and monomer is the name of the individual parts. So this polymer is made up of many parts. There are 4 of them. These are the names of what are called the nucleotide bases, symbolized by that. And at the right, I've shown a picture of one of them. If this were actually thymidyllic acid, it would have hydrogens here in place of those negative charges. RNA, close cousin of DNA, is long, almost always single-stranded, and it's made up of ribonucleotide monomers. There are 4 of these bases there. Here they're listed, these symbols. And the difference between the bases here and in DNA is just one. Here we have the base called uracil, here it's thymine. The difference between those, here's the uracil piece, and that doesn't have a methyl group at this position. Thymine does. Another difference between DNA and RNA, this is called deoxyribonucleotide because there isn't an oxygen at this position. This does have an oxygen at that position, so all the sugar part of these ribonucleotides are different. Okay, finally, proteins. Proteins are another class of these molecules. Long strands of amino acid monomers. There are 20 of these. They have the general structure here, and the linkages to their neighbors are between this nitrogen and that carboxyl group. Here's an example. This is glutamic acid. You might have heard of that, and any of you who know what MSG is, well, that uses the sodium salt of this stuff. Okay, now the terminology needs a little amplification. What do these terms mean? A lot of people, when they see these terms, they think, oh, that stuff, it's all the same. Oh no, it's not all the same. It's because these terms identify a type of molecule or a type of polymer. Each was different because it has a unique chemical structure and sequence of the monomers. To help you out, here's an example from language. We have a term called poem. What's a poem? Well, a poem is a literary composition. It's a type. But no 2 poems are the same. How are they different? Well, because they differ in their words and the sequence of them. It's like that with all these specific molecules. Here's an example like RNA. What is it? It's that. But no 2 of these are the same because they differ in their monomers and their sequence. Now, a clue about how these structures look. Professor Letsinger made a nice analogy in a piece he wrote, comparing these polymers to a train. Here is his picture of a train, bunch of cars. Cars are all similar, usually a little different, and they're all hooked together by this coupling between. Well, that's the analogy to the oligonucleotides. Here we have the oligonucleotide first one. That's an adenylate acid residue. That's what the A means here. And they're all hooked together in a long linear chain by these couplings. Much of this talk is going to be about how to make those couplings when you try to synthesize it. This one has those bases that we'll tell you about in a second, and they're a quick way to refer to that string would be to simply put the bases in capital letters there. Now, considering the structure of DNA, I start with single strands. This is a single strand of DNA that has 4 deoxynucleotide bases. So here's one, there's another, and so on. The single strands are what typically one tries to make if you're trying to make this stuff. But the business end of DNA shows up in this. This is the famous Watson-Crick model that has put out in 1953 by these 2 fellows. And it is still withstood the test of time, and we still think that that's a structure. There are some alternative structures too that we know exist. What's going on here? Well, this is one of the strands, and in detail, that's going to be sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, all hooked together by chemical bonds. That's one strand. And it's intertwined with another strand of the same thing, except that the ends are different. The polarity of this one is 5' on this end. That refers to a position of the sugar. So it goes 5' to 3', and this one goes the other way from 3' to 5'. In the middle, holding it together by some weak things called hydrogen bonds, the bases find each other. So here we see the bases sticking together in the middle of this double helix. Here are some alternative views of the DNA structure. Here we show a ribbon that takes some of the confusion out of it. The ribbons represent sugar phosphate chains. And in the center, a little bit more specifically, but schematically, we have the bases. The bases tie up with each other, always A with T and G with C. These are weak associations, so they can come apart. Here's a detail of that. The AT connection is through the hydrogen bonds. Those are simply attractions that are specific to certain atoms. And there are two with A and T. There are three that hold G and C together. And they go both ways. You can have GC left to right, or you can have GC right to left. They're okay with that. Here's another model. This is called a space-filling model, and it is constructed on the assumption that we've simply represented the atoms there that are present as filling out the space that we know the electrons of the atoms occupy. But you can still see the double-stranded structure going around here, and the bases pairing up with each other in the middle. Now, we get to how this works in the cell. We have in the cell, Watson and Crick, about 5 years after their famous structure, that put forward what they call the central dogma. I think that's very funny, actually. Central dogma. This is science, right? And we're talking about dogma. Well, what they are saying is that this is how it always works. Okay? And so the dogma is that there's information flow in cellular biology. And the information flow comes from the sequence of monomers in each of these giant molecules. That is the information. And there's something called the genetic code that it follows to read that information and to transmit it. There are three processes for these transmissions. One is called replication. That means the reproduction of DNA by copying itself. So whenever cells have to divide, that's involved, or in species reproduction, that too requires DNA to copy itself. The agency of doing this is an enzyme. We're not going to talk very much about enzymes today, but these are giant protein molecules that, I might say, oversee the processes of chemistry that go on between these molecules. This one is DNA polymerase. And transcription, that means the copying of one strand of DNA into a complementary strand of RNA. Mostly the RNA that's made is called messenger RNA, and the agency of doing that is RNA polymerase. Finally, translation. That means the reading of a sequence of messenger RNA in a ribosome to make a protein. But the sequence of amino acids in the protein is dictated by the sequence of bases in the messenger RNA. A little bit more about that later. Just to give you some idea, here's an example of one step of the process of replication. How DNA polymerase replicates a template DNA strand. Here's the template. That's the piece that's being copied. So we've already copied, making the new strand, the copy strand. We've already copied 3, and they're in place in the new strand, and we're putting in a 4th. This one, that's the monomer that's going in there. These 3 phosphates in a row represent a activated group. This group is with the 3 phosphates is ready to make a bond with its neighbor. When it does so, DNA polymerase causes this phosphate to make a bond with that oxygen. And so when it's done with that step, here you have the new bond, and you have a T, has to be a T, the others won't fit, coming in to associate in the hydrogen bond with the neighboring A. That's one step, and you can see how information, that is the nature of the bases, dictates what's going to happen here in reproducing this. Might add that one of the most famous footnotes in science comes from Watson and Crick's first paper proposing the structure of DNA. And the last sentence of that is something like this: it hasn't escaped our attention that this complementary base pairing that we're proposing suggests a mechanism for reproduction of DNA. It didn't escape their attention. What? Of course. It's really obvious once you see the structure, and that base pairing is what dictates all this that goes on. Okay, a little bit more about the central dogma, looking at the next 2 kinds of things, the transcription and the translation. Here we have a double-strand piece of DNA that's represented in a straight line. It has to unwind from the other strand, and when it does, if we reproduce this top strand, we're going to get an RNA which has this sequence. Since these 2 strands are matched up, their bases shown here are the same as the bases down here for the RNA. So the top strand has been transcribed into messenger RNA. The exception, of course, is that we have uracil in here in the place of thymine up here. So when it goes out from the nucleus where the DNA is, we manufacture the mRNA in the nucleus, but it goes out from the nucleus to the rest of the cell, where you have these amazing organs called ribosomes. And in the ribosome reads this code from end to end and makes a protein like this. The reading is of 3-letter words. Here's a 3-letter word, and if you want to know, well, what's the genetic code, it comes in triplets. Each word in the genetic code is one of these 3-letter words. And this 3-letter word over here, AUG, that tells the ribosome, put a methionine in the protein that you're making at this position. Here's another 3-letter word, and it says, put a threonine in the protein at the next position. So you read along, or the ribosome reads along here, and it puts the corresponding amino acids in the right place in the protein that it's making. Notice one other funny thing. Here's an example. Glycine has 2 different words that code for it. Here's a GGA that codes for glycine, and here's another. That's a GGG coding for glycine. Well, there's some redundancy in the genetic code language, and there are some amino acids that have 3 different words that code for them. Some have 2. Sometimes that can cause serious problems when you're trying to do synthesis of these things. Here's another important fact that you need to know. These complementary, if they are complementary, base or single strands, having bases that complement each other in different strands, can bind, that's called hybridization, to make double-stranded DNA. So here's an example that Professor Letsinger put out. He said, suppose I have a sequence that I have figured out, and here are 20 bases along the sequence that I figured out. That figuring out means that they sequenced it to make it. So if you sequence, you're going to figure out that sequence there. Now, if I make a piece of DNA that has complementarity to the center part of this, and I put them together in water at 60 degrees, these will tumble around and find each other. And when they find each other, they're going to lock into a double strand piece like this. And all of the complementary bases are going to be lined up next to its complement. So here's the AT, GC, GC the other way, AT the other way, and so on, all the way through this string. These bases are complementary to these bases, and they'll come in and stick at that particular position for that reason. Now, if you want to take them apart, you can do that too. All you have to do is heat it back up a little hotter, 95 degrees C, and these come apart and go to the single strand position. I bring that up because a lot of what we're looking at today is going to be sequence the DNA in some target organism, and then get in there somehow with something at a certain point that causes that thing to do something that you want it to do. This is how you get in there and do that. Okay, well, now we're ready to consider how to make this stuff. So starting here, you've seen this slide before, but now I want to emphasize that this is a tetranucleotide, and here's one of the monomers. Here's another. Here's another. Here's another. Well, how are they held together? Well, by this bond. The phosphate of one of these nucleotides has to make a bond with the sugar of the next one. And similarly, this has to make a bond with that one. That has to make a bond with that one. When you make those bonds, then you have those four stuck together. Now, you might be a little flummoxed by, what do you mean, stuck together? In what sense is that? Well, here's how chemistry operates in a real simple case to stick something together by making a bond. This is an elementary organic chemistry reaction that even Dave Ferguson knows this. Here we're putting this chemical together with that chemical and making this. Those are the names of them, which tell you not much. But here the way this happens is this thing, the methoxy group of that, comes and makes a bond at the back side of this carbon, and it boots out that bromine. Here's how it looks in the middle, and at the end you have a bond made here, and the bromine has been kicked out. It's called a leaving group, and bromine is a real cheerful leaving group. It likes to leave. Okay? And so, in what sense well, this is a new compound, and you can't get the methoxy out away from the butane because it's got a bond here. That's a strong bond that holds those two together. Okay, so now we turn to the subject of oligonucleotides and how to do that. Here, this is a little more complicated. First thing I'd point out is that you want to make a bond between this position, there's a carbon there, with an oxygen to the phosphate, or phosphorus of the phosphate. So you want to make that bond. That's the agent of doing it. It's called dicyclohexylcarbodiimide, which I won't give you a quiz about that, but it is called a condensing agent. It's very good at getting these two together. The way it works is this comes in, makes a bond with the oxygen of the phosphate, and that transforms this into a leaving group, which then this can come in and kick out like the bromine in the previous example. So when you get done, you have them linked together with
turning promptly. Because technical difficulties forced us to start the lecture 10 minutes late, we're going to extend the lecture 10 minutes today. So we'll officially stop at 11:40. We understand if you have a commitment and you need to leave at 11:30, Professor Wubbels understands that. But we'll officially end at 11:40. Once again, Professor Wubbels.
Thanks, Barb. Notice that the end I put down that this reaction is slow and messy. We need to talk about that. We'll talk about the slowness quite a bit later, but the messiness is understandable this way. First, I'm going to say, well, here's a picture of a reaction flask in which we have a solvent, and the reaction we just talked about is in there. The messiness of it is that it doesn't go to completion, so that you get about 95% of it making that product. But now the problem is, how do you fish that stuff out of the solution? Because you need to isolate it as a pure crystalline solid in order to take the next step. Well, that's called the workup and purification. What you have to do is get rid of the solvent. DCC makes that product, and that's in there. And you have a little leftover starting material, and then you're probably going to have a little side reaction products in there too. I'm not going to tell you about workup and purification, but I'm going to say that it takes a long time typically. So a couple days, maybe a week, that you have to spend working to get that pure stuff out of there. The reaction itself is slow. As I said, it takes about 2 days to run the reaction to close to completion. Professor Letsinger got a brainstorm about this time about a way to do this synthesis and avoid the messiness of it and the time-consuming workup. And it starts from something like this. Do you all know what that is? That's packing material, which chemists would call polystyrene. Well, actually, you probably know it as that too, except that in the labs, this stuff is called a popcorn polymer. And you can guess why. After the polymerization reaction, which starts with liquids, this stuff shows up, and it looks like popcorn. Well, you can grind up the popcorn into little tiny pieces, but that's what Bob thought might be a key to fixing the workup. So here's a schematic of what the popcorn polymer looked like. The popcorn polymers that we have as packaging material don't have any active group in them, so they needed an active group in there that they could run reactions on. So they tried to put a carboxyl group in there and were successful. So this is the recipe for making a popcorn polymer that he could use to do a certain kind of synthesis. You can see the chains here. There's a long chain that runs this way. Here's another one running that way, and another chain going this way. They're linked together because of the cross-linker. There's a little tiny bit of this in there, and that provides the connections between the chains that holds them together. And there are two of those I've shown. And in addition, one of the reactants is this that has a carboxylic acid group on it. And so they show up too, and so now this popcorn polymer has those things on it. The symbol for this is this. P stands for all the popcorn polymer, and that is the active group that is stuck on there. Of course, I've shown just one, but there are, of course, billions and billions of these in any sample. The way Bob thought to do it was that he would put the popcorn polymer in a funnel like this with a filter plate here. So grind up the popcorn polymer, put it in there. Now you run the reactions on this, and to put it in the vernacular, the good stuff always is what's stuck to the polymer, because it's made a chemical bond with the carboxyl group. Okay, so you can then filter this, and all that other stuff that we saw earlier that's in there that we don't want, all these side products and other things, they're still in the solution. They aren't bound to the polymer. So you can just wash them off. Brilliant, huh? And that saves 2 days or a week of workup and purification because it's stuck on the polymer. So then they carry out additional reactions, sticking more things on here, and each time afterward you just wash it off with solvent to get rid of all the other stuff. Well, that was a great idea. And so Bob thought, well, we better do a proof of concept here. So he did this. He made a dipeptide. That's a amino acid linked to another amino acid. I won't quiz you on all these chemical reactions here, but I assure you that they're simple little organic reactions. When you get to the end, what you have is this. So you put on one amino acid here on the group, and then you've added a second one. So here's your dipeptide. And now when you heat it up with hydrobromic acid, and it chops everything off, it doesn't chop the bond that you've made, so now you have your dipeptide. Letsinger and a postdoc named Milt Cornett did this in 1963, and about a month after they did it, they were chagrined to read in the Journal of the American Chemical Society that a fellow named Bruce Merrifield at Rockefeller University had done the same thing on a popcorn polymer. Well, that's called being scooped. So Bob immediately published his work. It had appeared 2 months or so in the same journal after Merrifield's. The irony of it is that 20 years later, Bruce Merrifield was awarded the Nobel Prize in Chemistry for developing the world's first solid phase peptide synthesis. Letsinger was ignored. Well, those of us in the Letsinger camp thought that was a great injustice. Marvin Carothers said in a published article, this was a great disappointment. We thought Bob Letsinger should have shared this honor, and I believe that's true. He should have. But he didn't get it. Merrifield continued on to do research on polypeptides made with this method, and Letsinger promptly turned his attention to his real aim, which was to try to make polynucleotides. So, did I go 2? No, I didn't. I think I want to do this. Here's the synthesis that he did about 3 years later of a dinucleotide on a polymer support, the same polymer support. See if I can get back online here. Again, I won't quiz you about the reactions that are here, but at the end, what you get is a dinucleotide, and at this point, you've made the linkage between the two. If you wanted to add another one, you could imagine taking this up here that has the same functionality as this and do another addition, and you can keep adding them in principle. Or if you wanted to stop, then you could put in this reagent, which chops off the blocking groups. This chops off another one, and here you have your dinucleotide. A couple weeks ago, Northwestern University was recognized as the site of what the Division of the History of Chemistry of the American Chemical Society called a chemical breakthrough. And they celebrated 57 years later this breakthrough discovery of Bob Letsinger. There weren't very many of these. There only been about a dozen in the whole field of chemistry over the last 30 years, so this is quite an honor. So it is commemorating the first solid phase synthesis of an oligonucleotide on a solid support. Joyce and I had the privilege of going to this symposium, and so here we are. That's a picture. Here are Joyce and I listening to the lecture. A couple notable people here. This man is the Robert L. Letsinger Professor of Chemistry at Northwestern. And this is the person who organized this, made the nomination of this development. She is a graduate of the Letsinger group as a graduate student. And that fellow up there is Fraser Stoddart, who won the Nobel Prize in Chemistry and is now on the Northwestern faculty for developing molecular machines. That was happened in 2009. So now the other problem I mentioned earlier, slowness. Here is the beginning of a solution to slowness. Letsinger and Lunsford and another fellow named Heavner, who was in his group, a graduate student, discovered that you could do these coupling reactions in a different way, and it was much faster. This used a thing called a phosphorodite. So this is phosphorus in the 3 oxidation state, which has 3 bonds to neighboring groups, and those are exceedingly reactive. So you can stick that together with this at the phosphate level, you get a phosphate, and you can do another addition here of replacing that chloride and get to this one. The beauty of this is that these take about 1 minute to run. So they're really zippy. It's estimated that these reactions go about 10,000 times faster than the coupling reactions at the phosphate level. And the beauty of it is that you can oxidize the phosphate that you get here to phosphate very simply. You just dribble in a little bit of iodine solution in water, and it takes it right to the phosphate. Well, this was a huge breakthrough. There were some problems, however. Bear in mind that what Bob really wanted to do eventually was to automate this, that is, to make a machine that could do these additions of reagents. Well, problem with the phosphochloridites is that they were too reactive. They needed to be cooled to -78 and put in an inert gas atmosphere to avoid bursting into flames. Okay, and so a chemist can do this fairly easily, but a machine really is challenged to do that. So it needed to be tamed somehow. The second thing is that the popcorn polymer had problems. We determined that it swelled, and when it swells up, it absorbs reagents, and those reagents then are in there, and they screw everything up from that point on. So those were the 2 big unsolved problems. Come then to this person, Marv Carothers. So here's a photograph of Marv from 2006 when he received the National Medal of Science Award at the White House. That's a big deal, by the way. So they recognized Marv. Well, so now, about 20 years earlier, Marv got to work. Oh, well, another story. In about 2010, my Christmas card that went to Professor Letsinger came back to me undeliverable. And so I thought, who in the world knows where Bob Letsinger would be now? So I called Marv up on the phone and I said, Marv, you know where Bob's address is now? Oh yeah, he knew that. So he emailed me that. So then when I looked up where Marv was at the University of Colorado, I found out that his office and lab was in something called the Jenny Smalley Carruthers Biotechnology Building. I thought, that's an amazing coincidence. Marv Carothers has his office and lab in a building named Carruthers. How did that happen? Well, the story is that Jenny Smalley Carruthers was Marv Carothers' wife. And they'd had a long life together. She died unexpectedly in 2006. And so Marv, owing to some activities I'll tell you about a little later, made a contribution of $20 million to Colorado to build this building. Well, 20 million wouldn't do it. They needed about 6 times that amount of money to build this building, but they did. This is an impressive building. 330 square feet of office and laboratory space in here. And they also got some programming in there. And here's a familiar face to some of you. This is Tom Check, who is a Grinnell College graduate, 1970. I had the privilege of teaching Tom Check biochemistry in 1970 at Grinnell. Tom went on to a distinguished career at the University of Colorado. And so here's Tom on about the year 2000 when he became the president of this huge medical institute that funds fundamental research. And he did that for 10 years, and then he came back to Colorado as a chemistry professor. And so here's a picture of Tom and the leader of this institute that was created. They have about a little over 20 faculty that are in this biofrontier. That's an interdisciplinary group. And we have a view here looking out to the west from the Jenny Smalley Carruthers building, seeing other buildings on the campus. Here's a picture of the Carruthers building. The red arrow indicates the headquarters of this Biofrontiers Institute, which Marv Carothers created there. That's an amazing set of accomplishments, I think. So, Marv went to work on these problems. First problem was curing the popcorn polymer problem. He did this in a most ingenious way. He said, let's take silica. You probably know silica, that's sand, right? This is very finely divided, highly purified sand, and those will be actually tiny particles, but relative to the size of these molecules, they're huge. They're especially adapted to let liquids flow through them. Very desirable feature for what the purpose was. So he derivatized the actual beads this way and puts on a carboxyl group, which is its handle. Then you can do these chemistry reactions. You can stick one on there using DCC to stick it on, and you can put it together with a phospholorodite. This was a chlorodite in the first case. That means that they had to keep it cold and under an inert gas atmosphere, but it worked fantastically well. And they were getting yields of this on the silica beads of about 98% to this product. And it occurred in about 2 minutes. Wow, pretty impressive, right? We still have to tame the chlorodite reaction. You can't have that going that, having those requirements. So here's how he tamed this. This isn't a very good slide for showing that, but it shows some of it. Here, when we put that together, now the phosphate is no longer a chlorodite, it's an amidite. You do that by reacting the chloride with the secondary amine shown here, and that makes it into an amidite. Another beauty of this is that this thing is stable in the atmosphere, quite stable to water, so you don't have those problems. And when you put that in with this weak acid, tetrazole, it will put a proton on that nitrogen and pop it off as a leaving group. So you can make now the reaction with this hydroxyl group here and stick those two together to make the phosphate. This was also very peppy, about another 2-minute reaction, and that's done. And then you can oxidize it to the phosphate, and then you can continue on. If you want to take the blocking groups off and get your product, you can do that, or you can do another cycle. So each one of these cycles here would represent adding another link in the chain. This was, if I say so myself, this was really impressive chemistry. Both the learning to use a different solid support and also taming that phosphate coupling reaction. So, Carruthers succeeded there. This was about 1980 when this was happening. At this point, a career-changing event occurred. A group of scientists got together and decided to create a new company which became known as Amgen. And Carruthers was one of the founders. That's probably where he eventually got his $20 million to donate for the building. But one of the aims of Carruthers and Letsinger was to develop a machine that would do this. They now had the chemistry that would work in a machine. So they built a machine. Amgen, they decided, was really the wrong place to put a company to build machines. They wanted to do biotech stuff with that, and the machines did somewhere else. So he created another company called Applied Biosystems with a guy from Caltech named Lee Hood, and some venture capitalists. They created this company that was going to be to make machines, including the world's first DNA synthesizer. Okay, so they worked for 2 years to do that, and December 1982, this fellow shows up in Boulder with the world's first DNA synthesizer. Here's a picture of Bill Efkovich, former member of the Carruthers group, and Bill's the one who delivered it and set it up in the Carruthers lab at Boulder. So now we're into using machines. This worked, and here's a synopsis of what's happened in the automated synthesis of oligonucleotides by Carruthers chemistry. Right away, these became available, and you could make DNA oligonucleotides of 20 to 30 bases in length in 1983. This is a little more complicated to make RNA this way, but that was done about 10 years later. This company, and then many others, when the technology was patented and then licensed, many companies are now making oligos, and they have improved things so they can make them up to about 100 base pairs. They also developed what are called analogs. These names represent different kinds of polynucleotides that have different atoms in their backbones. That's advantaged because you can now do a lot of things in drug development when you've got modified reactivity in the polynucleotides. By this last decade, you could do these syntheses of up to about 300 base pairs in length, and then you could make much longer ones by some tricks involving ligase enzymes and polymerase enzymes. And last about this technology I'd like to take a glance at, which is this. Carruthers and others have figured out how to do this amazing thing. They modified an inkjet printer to work in these machines to do a solid phase synthesis of the DNA on glass plates. And this is pretty astounding. There are 12 chips here on one glass wafer about 6 inches square, and there are 244,000 DNA sequences that you can make on one chip. So for one run of this, if you went to completion, you'd be making about 3 million different DNA sequences on this chip. And the yield of just the good stuff in each case is about 45%. That's not too bad. There are a bewildering array of things you can do with this now, and people are working hard to employ this. Well, I think there's going to be another revolution here. But let's take a look at some applications. As soon as they started the Amgen company, they realized that there was a very important possibility for research. It involved this. Erythropoietin is the human protein that is synthesized in the liver, and mostly the liver, and it stimulates the production of red blood cells. Quite a few really sick people need to have their red blood cells stimulated. So there was a big prize at the end of this if they could learn how to make erythropoietin. Well, they did learn how to do that, and the story involves synthetic DNA. The problem was that while the protein had been discovered and isolated at the University of Chicago, they didn't know what the DNA sequence was that coded for it. So they decided to find it out by trial and error. So there's a lab of 10 chemists that were set up by Amgen in Boulder for Marv to oversee. And these guys set about making short pieces of DNA to hybridize with their suspected regions of the piece of DNA in the human gene, and so they started doing it. Well, for 2 years of trial and error, the Boulder chemists made about 700 of these short sequences. There were probes to go in there and try to stick on certain parts of the human genome, which then, if it stuck in the right place, they could snip out with enzymes and get the gene that is coding for that. Well, they finally succeeded in doing that, and then there was a quite dramatic effect of these. They were working with a mouse model first, and when they injected the protein that was made by this method, recombinant DNA in Chinese hamster ovary cells, the mice that they injected with this erythropoietin died. And what did they die of? Their blood vessels were so clogged up with red blood cells that they died. And this was a dramatic indication that they had the right sequence. So they started producing this stuff, and of course you don't want to kill somebody by putting it on. So they learned how to do it. Remarkable thing is, here's a global estimated sales of this or things like it, estimated at about $10 billion a year last year. Oh, that's amazing, because that's 30 years after it was first marketed. So it's all off of patent now. Here's a way it works with a patient. This is an anemic patient getting a blood transfusion, and it boosts it up short, and then it falls back down. But when they put intravenous epoietin in, then this is what happens to the red blood cell count. Truly amazing medicine. So that protein that they made was not exactly the same as human one. It was a little bit modified. That was a fusion protein. We need to know a little bit about that. What's a fusion protein? It is two proteins in one strand of amino acids, but they're made from recombinant DNA on these Chinese hamster ovary cells, and the genes are transcribed to messenger RNA and translated into one protein with two active regions. common link-up is between an antibody protein and a protein with useful biological activity. Flesh that out a little bit. Here's how the Chinese hamster ovary cells work in recombinant DNA. First, you have to have your synthetic DNA, and this represents that. It looks real short here, but actually it could be very long. And it has uneven ends. This double-stranded DNA has what the scientists call sticky ends. They're sticky because those bases that stick out here on this end are going to want to match, and that's not an accident. They're made to match the sticky ends of this thing called a plasmid. So when the sticky ends find each other, it zips up to that, and there's an enzyme called the ligase that will create the chemical bonds to stick this right in that thing. Plasmid, those are extranuclear DNA that a lot of bacteria have, and that can be isolated, and these are now used routinely. There are over 100 of these that are available. And the trick is now you want to do what's called transfection. You want to put this recombinant DNA molecule into your Chinese hamster ovary cell. When you do, and it's successful, it looks like this is the DNA of the Chinese hamster ovary cell, and this didn't work, and so you want to get rid of this, but there are biochemical tricks you can play to get rid of that completely and make only this, and then you can do a process called cloning of that cell and get millions of copies of this one that has the external DNA in it. That is then the reaction flask for making the recombinant protein. Here's an illustration of the end result, or something like it. Here we have two joined together. That's the piece that we want to do something. And this is a antibody protein that it was put next to. There are two of these. They just like to get together. You can stitch them up with this chemical bond. And so the fusion protein is that whole thing. And you might say, well, how does that work? Well, here's an example of how it might work. Here's one that was developed to treat the COVID virus. The COVID virus infects things because it has a thing called a spike protein. And the shape of that spike protein is such that it likes to stick to these receptors on human cells. And if it sticks to them, then you're going to get sick. It'll infect the cell and be successful. So the antibodies that are specially made so that they like to stick to the spike protein, if they're in there, they'll come and tie up the spike protein like this. And that'll prevent that from sticking down, joining the receptor on human cells. I told you that I was fascinated by how these diagnostic tests for COVID worked. Well, this slide is pretty complicated, so I'm going to skate over it fairly quickly. Here we have viral RNA. So if you swab your nose and you come up with some of this and it goes into the test solution, that virus is going to be in there. This is just a short piece of it that we're looking at. The DNA that's synthetic is here. That requires this as a primer to come and stick to a certain region that it matches on the viral genome. So when it does, it tells this enzyme called reverse transcriptase, hey, come in here, start running down the line to make DNA from here. This is a start point. That's why this thing is called a primer. So it does that, and you get double strand here. This is now complementary DNA that is made from the viral RNA. You can break those apart here by heating it up, and then you can put in more primer. And now this is shown for just one, but there will be, of course, millions of these going on in the tube. And then through some tricks that would be too tedious to outline, you put on a probe which has a sequence of bases that match a certain region here that we know for the RNA, and that has on it a fluorescent molecule, next to it a quencher molecule, so it won't fluoresce, that is give off light, while it's next to the quencher. But when you break them apart and put on more polymerase next to that primer, it chops that off, and so the fluorescent piece comes now into the solution, and you can detect that by fluorescence. How's that for an explanation? It is really amazing. These things can detect, a very small amount of the virus in your nose with absolute certainty, and so it's specific and fast. It's just pretty amazing. So, as you know, I'm hunting for places where synthetic DNA is involved. Here's another one. The Moderna mRNA vaccine is a really complicated piece of chemistry. This piece here is the important piece. That's the coding region for the spike protein. So that's RNA. It has to have these ends here called untranslated regions, and those are basically transfer RNA molecules that are hooked on there, and then you need this kind of a tail on the 3' end and that kind of a cap on the other end. So when you make it, and you want to get it into these things called lipid nanoparticles. This has represented probably 10 or 20 years of research to create these lipid nanoparticles. They're really hard. But this circle actually is a three-dimensional thing. This will be a sphere, and you'll have the messenger RNA, this, inside there. And this bunch of lipids loves to, when it gets in your bloodstream, sit down on the lipid layer called a membrane of your cells and inject this into the cell. When it does that, then your cellular machinery says, hey, here's a new piece of RNA, let's make copies of the protein that represent, and that's the spike protein. So it makes, your own cells make the spike protein from that RNA that's in this vaccine and diffuse out into the bloodstream, and then the antibody makers say, hey, there's a strange protein here, let's make antibodies to that protein. So that's how you get immunized by the vaccine. I should have said, making RNA from DNA is a lot simpler than trying to make RNA directly. And that's why synthetic DNA is what they use to make the synthetic RNA. I'm just touching on the surface of this. So here are 5 different research areas that are really hot right now that all involve synthetic DNA or RNA. I'm not going to go into all of them, but in 2022, there were 282 of these kind and that kinds of drugs that were in clinical trials. That represents a flood. Okay, pressing toward the end, here I saw an article in Barron's magazine, that's a financial magazine, about what's going on in big pharma and what it means for investors. What's basically happening, big pharma is changing. This isn't going to be big pharma for very much longer. The reason is that they're getting into big biotech. Here's another distressing fact, 2 of them. This year, 2% of prescriptions were for these things called biologics that I've been talking about, but 37% of the drug spending was for these. And this 5-year period, only 9% of the newly launched drugs, that means newly patented, without health insurance costs $150,000 a year for a treatment. In this 2-year period, 47% of new drugs cost that much money. That's pretty daunting, right? And the reason the drug companies can get away with this is that they target these fusion proteins that they're making to really serious diseases. So people can't live without them. So that's why they can charge those kinds of prices. Here's what's happening in the industry. Here are about a dozen of the biggest pharmaceutical companies, and what are they doing? They're changing their corporate strategy big time. They're divesting. So each of these out arrows represents one of these big companies getting rid of something that they do. Why are they getting rid of those things? So they can invest in biotech. So it's all changing rapidly. And finally, here's something I saw in the news magazine of the American Chemical Society in January. That headline caught my eye. And then this one was also in there, shorter one. These companies here are already in the biotech business, but they're expanding their capacity to make oligonucleotides. And that's not a small investment that they're making. You can read about them here. Here's one you know about. They already are a successful company, but they're expanding by buying a DNA synthesis and amplification company for that amount. And BioNTech, the German group that combined with Pfizer, they're buying another one that specializes in these kinds of things for about $700 million. You hear the earth shaking? Right? It is. So that is my personal story about synthetic DNA. As I reflect on it, I thought, holy cow, here 60 years ago I was watching these guys slogging through the swamp trying to make synthetic DNA. They weren't making much progress, but now it's a mature chemistry, and the world is changing right under our nose because of this. So thank you very much.
Thank you. Thank you, Professor Wubbels, for helping us to understand how research into DNA has impacted not only our health, but almost every aspect of our lives. Thank you so much. It's a fascinating story. We look forward to seeing everyone next Wednesday, March 29th.
Is it true that I finished 5 minutes before the appointed hour?
You did.
And
you did very well.
I'd like to offer the opportunity to ask questions. So
would you like questions?
Some of you will be
3 minutes for questions.
brave enough to ask some questions.
I will bring the microphone if you'd like to ask a question. No? Bill?
Yes. As far as like making Dolly the sheep, where do we stand in terms of cloning on an international scale?
I'm completely ignorant about cloning. I don't know how it's done. I do know that it is done on sometimes a surprising scale. We have whole herds of sheep now that are cloned. I know that, but I don't know how they do it. I don't know what the implications are. I don't think it has anything to do with synthetic DNA.
So last year, one of the great advances, celebrated advances, was using artificial intelligence to discover the folded protein, the structure of the folded proteins. Have you seen much evidence of the use of artificial intelligence now to be working on some of the upcoming projects that you listed?
No, good question. The use of, I think it's probably not a accurate use to say that discovering how to predict protein folding was an artificial intelligence project. It used a lot of computing power, but it did require a lot of brain power to to figure out the algorithms you need to predict that thing. I mean, the number of flops per second you need to do that are just incredible. But there's a big problem with artificial intelligence and scientific discovery. It's something that humans can do, a few selected ones, but artificial intelligence is pretty dumb about that. But there are certain areas of science where this is going on now, where people are trying to use artificial intelligence to facilitate things. One thing it can do, artificial intelligence is a amazing adder. It can add things in ways that are tedious for humans. So when you have a really, really big addition problem, which is basically the kind of computing that you need to work here to predict what's going to happen, they're pretty good at that. Discovery, now that's another thing.
Okay.
So Dave, did we answer all your questions?
Oh.
I guess so.
Okay, well, we will look forward to seeing everyone next Wednesday when Grinnell College Professor Stephen Andrews will pose the question, what do baseball and two contemporary novels have in common? Find the answer in his presentation, No Runs, No Hits, No Errors, Marilyn Robinson's Deferral of Perfection from Gilead to Home. Thank you so much, Professor Wubbels, and thank you all for coming.